We are very excited today to have with us Dr. Brian Corrie. He is the Technical Director of iReceptor based at Simon Fraser University in Canada, and also the Technical Director of iReceptor Genomic Services. Today's talk is going to be iReceptor and the T1D, or Type 1 Diabetes AIRR Consortium. And a little bit about him. Again, as I said, he's the technical director for both these projects and the co-lead on the AIRR Common Repositories Working Group. He's a member of the AIRR Standards and Diagnostic Working Groups, and he has a background in computer science with a focus on data visualization, computational science, and scientific collaboration. And we're really pleased to have him here and to kind of walk us through the what iReceptor and AIRR are all about and how they intersect with the type 1 diabetes community. Thanks, Brian, for joining us.
Thank you, Monica. Can you hear me okay? Everybody can see my screen.
Perfect.
Since we have a pretty small group online here, I think feel free to jump in and ask questions as we go. I'm going to cover a number of topics. Let's just get going here. There we go. So I'm going to talk about the AIRR community briefly, and then dive into what the Type 1 Diabetes AIRR Consortium is all about, and then talk a little bit about how iReceptor works in the T1D context, and then we'll just open it up for questions and discussions.
Sounds great.
All right, so onto the AIRR community. So I think many of you probably know, but maybe I'll just kind of talk about this in a little bit of background. So I think everybody knows next-gen sequencing over the past 15 years has caused this explosion of data sets, and in particular in the immune response. There's approximately 10 to the 13 possible antigen receptors, 10 to the 8 cells per person at a single time point. When people sequence these data sets, you get on the order of 1 million sequences per biological sample. You get more than one time point per subject. You have many subjects, many studies, many labs. So you're getting an enormous amount of data is available. The AIRR community was formed in 2015 to try to tackle this problem. They recognized that this data deluge was coming, and they got together to talk about creating standards for generating, annotating, and storing these high throughput AIRR-seq data sets. And the goal is basically to be able to share the data effectively, which we'll talk about in a second. So the AIRR community works through a set of working groups. I'm not going to go into any detail into any one of these in particular, other than to point out that these groups have been meeting since 2015, so basically on the order of 10 years, working diligently to establish a set of standards. And the way those standards are formed are basically by the working groups coming together, putting forth a standard. There's 3 standards here that I'm talking about. The first was in 2017, came out of the Minimal Standards Working Group. There's a data representation standard and a query API standard for querying and finding AIRR-seq data. Now, these standards are published in papers, but the interesting thing is that the standards, when you're published as an AIRR community standard, those standards are ratified by the full AIRR community. So a vote is taken for the entire AIRR community. So tens of people, or if not hundreds of people, are supporting these standards. The AIRR community is open, and we're always interested in new members. So if you would like to join us, please reach out. You can find contact information on www.airrcommunity.org. One of the big outcomes and kind of the focus of what we're talking about today is what's called the AIRR Data Commons. So basically, the AIRR community came together and said that in order to share these data sets, we need to have a set of data, a set of repositories that are standards compliant. This will allow researchers to basically search these repositories across study metadata, study time points, subject age, sample, tissue, disease state, et cetera, et cetera. And having a set of standard repositories distributed internationally means that you can scale out to billions of sequences across tens or hundreds of repositories potentially. And this also allows you to have data curated at your home institution under your local data policy, if that is important to you as a researcher. Sorry, my PowerPoint just died. Give me a second here to find that document again.
No worries. Yeah, so I'll just sort of fill in a little bit here. The reason that Brian and I even met is after we did the first data-driven D challenge with the sugar science and a collaboration with DKNet, we realized a data set was missing. A TCR and BCR data set was missing for the type 1 diabetes community. And so a good friend pointed out that Felix Braden had a really great standardized and harmonized database. And when I reached out to him, he was a friendly Canadian scientist, and he agreed to help us create a vertical within the AIR Data Commons accessible through iReceptor, where we could house and build a large TCR, BCR database that was particular to type 1 diabetes, and that we could, Sugar Science would reach out and gather the community so that they would deposit their data. And of course, Brian Corrie was his sort of right-hand man there, a scientist who was actually running the technical part of this iReceptor AIR Data Commons, and he also joined the crew. And so that's where we started to build and gather scientists like Aaron Michaels and Todd Brusco, who were two of the first scientists to deposit their TCR data, and then we grew from there. So it's been really a great synergy with the AIR Data Commons, both Felix Braden and Brian Corrie. So thank you both for that. Just a little backstory while you pulled those slides up.
Thanks, Monica. Thanks for filling in.
Yeah, I had to jump in. Didn't want that airspace.
Yeah. So this philosophy of the AIR Data Commons really is about making AIR-seq data fair, so findable, accessible, interoperable, and reusable. So researchers really need to find data and federate that data across studies so that they can basically use data between studies to really, the goal is to drive new insights. And this is true of all disease states, but in particular, as Monica mentioned, is the goal of the T1D AIR Consortium. So the AIR Data Commons is actually quite large. There are over 5.2 billion annotated TCR and BCR sequences in the AIR Data Commons. This is the history of the growth of the AIR Data Commons. We've been around and in production since 2018 with a significant amount of data. And in the early days, basically, we curated data sets a study at a time, and we had these small increases. More recently, we've been seeing kind of large incremental increases as research groups do very large sets of studies over time. You'll notice that there are a small number of fairly large repositories, so the different colors represent the different repositories, but they are international in scope. We have repositories in Germany, Canada, the US, etc. So now I'm going to go on to the T1D AIR Consortium. But before I do that, I'm going to just talk a little bit about the power of disease-specific initiatives, which is what the T1D AIR Consortium is. So it is a specific consortium that's targeted type 1 diabetes as a disease, recognizing that sharing data is really, really important to get a better understanding of that disease. The first instance that we saw this was with COVID, of course, and you're probably not too surprised. So in early 2020, studies started producing COVID-19 data. The AIR community in March of 2020 made a call for sharing of AIR-seq data. And over the period of about 18 months, the AIR community basically came together to curate over 1 billion annotated sequences from 27 studies, COVID-19 studies. So that was a pretty significant effort. I'm not going to go into this in too much detail, but this is basically a timeline. Important things to point out is that the first preprint with a COVID AIR-seq study was by Nielsen et al. And within about a month, we had that data up and available in the AIR Data Commons. And the COVID-19 data sets came pretty fast and furious, as you might imagine. And over time, we basically curated a bunch of data. And that COVID-19 curation continues to this day. But that really initial, over that first 18 months, really sparked very fast data growth. So this is where the first COVID-19 data was available in the AIR Data Commons. That kind of orange slice through the graph is the COVID-19 data from the repository that we ran. So that's the data that we curated. And essentially over that 18 months, you had this billion sequences, give or take, added of COVID-19 data that was available to the public. So the T1D AIR Consortium is kind of a similar initiative to that. As Monica mentioned, it was kind of initiated through the Sugar Science and iReceptor, talking about type 1 diabetes. We started talking to Todd Brusco and Aaron Michelle at the University of Florida and the University of Colorado, respectively, to try to basically coalesce the community of T1D researchers to sharing their data. And to that end, we created the Type 1 Diabetes AIR Consortium. And basically the mission of that consortium is to enable the sharing of type 1 diabetes AIR-seq data to better understand disease and disease progression. We recently published in October of 2024 a paper about this. You can see that on the screen. And this presentation will be available for people so you can kind of find out more details. One of the key things that we did with this project is basically created a curation process and protocol. And essentially what we do is we work with research teams that have data. So the collaborators in the AIR Consortium, the T1D AIR Consortium, and they work with essentially our curation experts from an iReceptor perspective to curate data into the T1D repository. The important thing about the T1D data in particular, so all data in the AIR Data Commons has rich study subject biological sample metadata. So you can, the data that describes the study is very rich and very comprehensive. One of the interesting things about the T1D data is it's one of the first large sets of data that has subject HLA being very important in T1D. But in addition, it has very rich VDJ sequence annotation. Some of it is paired chain. So you've got the heavy and light chain or the alpha and beta chain for the BCR and TCR respectively. We have epitope reactivity data. So we know which receptors bind to, in some cases, to which epitopes. And in the T1D data, we're approaching 400 million sequences from 9 studies in the AIR Data Commons. This is a open consortium, so we do invite people that are interested to, if you're interested in collaborating, to please reach out to the T1D AIR at thesugarscience.org, and we can fill you in. Monica will fill you in on kind of the consortium and how it works. We meet every 2 weeks, I think. Monica, is that right? I can't remember the cadence actually.
Once a month.
Thank you.
The third Tuesday.
Thank you. So this is just a little bit of the history of that sharing. So as we mentioned, we started this with Aaron Michelle and Todd Brusco. And so our first 2 data sets essentially were data sets from their group. So we created the repository in 2022. We added those 2 studies to kind of prime the pump, so to speak. And in parallel with that, we were working with Nina Prack at the University of Pennsylvania. She was doing some work with HPAP data. And so we had 3 data sets in the T1D repository fairly early on. At the same time, Aaron and Todd were looking at grant opportunities. They put in a grant proposal to the Helmsley Trust, and that grant was awarded in 2023, and that really sparked the growth and the addition of new data sets. So over time, we've been working with collaborators within the consortium, adding more and more data sets. And that will continue. Certainly the Helmsley grant has another a year and a half left in it, so we can expect to grow the T1D data and the AIR Data Commons significantly. I mentioned a billion sequences in COVID-19 earlier on. So currently, type 1 diabetes is the second largest disease condition available in the AIR Data Commons. And that is almost completely due to this T1D consortium initiative. I'm now going to talk a little bit about iReceptor. Oh my goodness, it's only 15 minutes.
That's okay.
I'm gonna be done really quickly. I thought this was
That's
gonna go a
fine.
lot
We can
longer.
have some conversation. It's great.
So what is iReceptor? So I've talked about repositories and data commons and databases and data and things like that. And I think we all know that having data in a repository or data in a data commons is great, but that data is often very, very hard to use. And the goal of iReceptor is to solve that problem for the researcher. So iReceptor is a web-based platform for finding, searching, and analyzing AirSeq data. iReceptor basically searches the data in the AIR Data Commons. So the AIR Data Commons is this distributed set of repositories. If you had to query each one of those independently and download data as a researcher, that would be very, very challenging. So iReceptor does that on behalf of you, the researcher. We've been in production since 2016, had a substantial amount of data since 2018. The iReceptor paper was published in 2018. This on the right hand is basically a citation graph. The iReceptor paper is essentially here. Actually, I'm going to put on my laser pointer here. So this paper here is the iReceptor paper. So these are all papers that have cited iReceptor as a place to find and store data. The x-axis is time, the y-axis is number of citations on a log scale. And you can see iReceptor paper itself is pretty highly cited. These green dots are all actually COVID-19 papers. So the COVID-19 papers are very, very highly cited, of course, as you would expect. And they all cited iReceptor as a place where they got or they stored their data. One of the interesting things I'd point out is these orange or orange-ish dots. Those are all machine learning papers. So those are all papers that are citing iReceptor as a place to find and get data to train and or compare data for ML purposes. So a little bit more about what the iReceptor Gateway does. As I said, it's a web portal that allows you to do interactive data discovery, exploration, and analytics. And it's really hiding the complexity of all these queries you might have to do to all these different databases. The AIR Data Commons is this distributed set of repositories around the world, and those are developed by the community of AirSeq data users, and they are all AIR standards compliant. So by having a set of standard compliant repositories, the iReceptor Gateway can send data queries to those repositories and federate the data and present it to you in a easily consumable way. In addition, so that's just showing the kind of queries going out to the repositories and data coming back. In addition, once you're in the iReceptor platform, you can actually do some fairly advanced analysis on the data within the platform. So you don't have to download the data to your desktop and do some offline data analysis. These data sets can be pretty large. And so the iReceptor Gateway manages some of that job management and analysis. And I'll show some details of that in a minute. One of the things that we've also developed through the iReceptor project is what we call the iReceptor Turnkey. It is basically a software stack. So if you want to store your own data in an AIR compliant data repository, we have some software that you can download and install on a server or a virtual machine in your lab and store and manage your own data in an AIR compliant fashion. And in fact, all the repositories that we run are actually using that software, of course. Some things to point out in terms of some of the capabilities. It isn't just about AirSeq data and in terms of the BCR and the TCR VDJ annotation. We have cells, paired chains. We can store gene expression, and we can store epitope and antigen specificity of the receptors as well in the AIR Data Commons. And you can analyze and find those through the iReceptor gateway. And then we've integrated some open source community analysis apps into the iReceptor platform as well. So this is what the iReceptor gateway looks like. So we're getting down to the nuts and bolts. It's basically a web portal, as I said. Anybody can get an account. If you want access, you simply go to gateway.ireceptor.org, click on the create an account page, and you will be off and running. I did mention that iReceptor was heavily cited. iReceptor is actually heavily used as well. This is basically the number of users that are added per month since beginning of 2020. You'll notice this really big spike. That's when the first COVID data was made available in the AIR Data Commons. So we saw an enormous amount of interest in the platform. We had over 70 people apply for an account in that month alone. And we're averaging on the order of, over the last 2 years, about 250 new users per year. And we are getting a significant new users every month. In fact, the last 2 months, so in February and March of this year, other than this COVID month, we've had the most users requesting new accounts on the iReceptor gateway than we've had other than that one COVID-19 month. So
Brian, can you,
is,
oh, sorry, can you parse that out to see if it's how much interest there is in the T1D portion?
that is a little bit challenging because when users use the gateway, when they send queries, they're sending queries to all the repositories.
Okay.
So I mean, it's possible to dive into it a bit, but it's a little bit challenging. The best way to see it is how much data is downloaded. I actually haven't done that recently, but that is possible because people basically, once you find data, and maybe I'll talk about this in a second, quite often, if you can't do all the analysis you want to do, basically you download the data that you find that's interesting. So
yeah, I mean, it might be confusing also because as they may be looking at the T1D TCRs, but then they might be kind of poking around at some of the other data to sort of cross-reference, other data sets. So it's really, that's one of the great advantages of this repository is that it's very standardized, but it's also very harmonized with other data sets, correct?
That's one of the things we like to point out. So quite often researchers, when they do their studies, if you're doing a T1D study, you might have a bunch of T1D subjects and you might have some T1D controls, right? So non-T1D subjects, for example, or healthy controls. The AIR Data Commons has a significant amount of healthy control data, right? So if you want to study healthy controls, then you might still do healthy controls in your study, but you also have other healthy control data that you could access through the gateway. Actually, maybe that'll become a little bit more clear. So once you log in, this is essentially the screen you see. This is the landing page. Just some things to point out. I mentioned there's over 5.2 billion annotated sequences, 59 diagnoses, 45 tissues, 6 PCR targets, PCR target being IgH, IgK, IgL, TRA, TRP, TR delta, TR gamma. 91 studies, 9 repositories. So it's a pretty large and rich data set. Both clone and cell data is a little bit newer for the AIR Data Commons. So we have much less clone data and single cell data in the AIR Data Commons, but that is growing as well. When you do start to browse things, you can choose to go down one of 3 different types of pipelines or workflows, so to speak. So you can look at sequence, clone, or cell repertoire metadata, and I'll talk about that in a second. One workflow that we find a lot of researchers, a lot of people are interested in the junction or CDR3 amino acid sequence because that is, is very prominent in epitope and antigen binding. So a lot of people are interested in just searching for CDR3. So it's very easy to actually just put in a CDR3. If you find a CDR3 of interest in your study from a specific receptor, you can basically put it into the gateway, hit search, and it'll search all 0.5, 0.2 billion sequences and give you back an answer in 30 seconds or so. So that's a very, very significant capability for this platform. This is what the metadata screen looks like. So on the left here, we have basically the filters that you can filter across. You can filter by study, filter by subject metadata, filter by sample metadata. Some of the things you can search across is disease diagnosis, age, ethnicity, sex, organism, et cetera, et cetera. In this case, I've basically done a metadata search for subjects with a T1D diagnosis. So here I've put type 1 diabetes as my my disease that I'm interested in. It tells me that that's my currently active filter. We have 69 million sequences from 8 studies, 606 repertoires. And those are all subjects with T1D. So there are basically about that many more sequences available for the healthy controls from these studies. So those are just the subjects that have type 1 diabetes. So you can see the study group in this column and the disease diagnosis is all type 1 diabetes for this. There are no healthy controls in this search. So what this search found is 69 million sequences across 8 studies, across 9 tissues, 3 PCR targets. Most of the data is TRB, so T cell receptor beta chain. It's very easy to start diving into sequence cells. So up here you have kind of we're on the sequence workflow. You can go into the clone workflow or you can go into the cell workflow. So we don't have a lot of single cell data from the type 1 diabetes as of yet. That's starting to grow. I'm going to go down the single cell kind of workflow path. So excuse me. So this is the cell search page. So basically what you see here is you've got the, in this case, this is a TCR, sorry, an IG data set that I'm looking at. You can see there's the heavy chain and the light chain. What we're showing here is the V gene and the CDR3 for the heavy and light chain. You also get the gene expression data through the gateway. And then one of the things you can do is, again, you can download this data if you're interested in exploring it on your own, kind of offline. But you can also provide or apply some well-known community types of open source algorithms to these. We have 2 single cell data analyses built in at the moment, Conga and Cell Typist. If you're familiar with single cell TCR, PCR data analysis, you're probably familiar with those tools. Basically what you can do is submit an analysis app, and that will give you after some processing, and these jobs can take a long time, but they're doing a significant amount of processing. You'll notice that we had 5 samples in this original data set. We get 5 different analyses. And in this case, what Cell Typist does is it actually infers based on the gene expression, the type of actual cell that each of the cells in this study are. So you essentially get a inferred cell type from each of the repertoires. So these are basically 5 different subjects in this study. So those are the kind of the nuts and bolts of the workflow on the iReceptor gateway. So I'll basically stop there and open things up for questions. I'll just maybe say a few more things. So really, the Type 1 Diabetes AIRR Consortium goal is to create this, essentially this network effect where we attract more data and more repositories, trying to make this the go-to place to find and analyze combined AIRR-seq and single cell data. And certainly from a T1D perspective, that's what the goal of the AIRR Consortium is. So we want to grow that T1D sharing, but also extend that sharing to other diseases, either other autoimmune diseases, but it's fairly critical to infectious disease vaccine development. cancer immunotherapies. All areas would benefit from the same spirit of sharing. And the iReceptor gateway is more than just an interface to the Air Data Commons. I kind of think of it as more of a platform for doing a data mashup. We're trying to bring together data from other relevant sources. So we link out to epitope databases. We link out to kind of the gene databases for the gene expression. On our gene page here, you see these are all links. So basically, if you click on these links, you basically go out to the definition of what that gene is, right? So it's not just data about the Air Data Commons, but we're linking to other types of data through the platform. And then to participate in the consortium, as I said, reach out to the T1D AIRR at thesugarscience.org. If you want to go to the iReceptor gateway, go to gateway.ireceptor.org. If you want to contribute or explore the Air Data Commons in general, please send us an email at support@ireceptor.org. And I'll just mention the collaborators and funders. First, the T1D AIRR Consortium. We also have a current NIH Air Knowledge Commons project. Our collaborators on the past CIHR EU Horizon 2020 iReceptor Plus project, and my colleagues in the Air community, and our funders, both past and present. And they're all stopped.
Thank you, Brian. That was really great, a great walkthrough. I guess I would just I have a paper, I dropped a paper in the chat. A recent paper out of Stanford from Scott Boyd and co. Maxim Zaslavsky was the first author, but also Anshul Kandage and Scott Boyd were the primary PIs. And it's really interesting how they, really took a look at sequencing and interpreting BCR, TCR genes and using them in context of creating a single diagnostic test for many diseases, including type 1 diabetes. And they basically took blood and then they used their machine learning for immunological diagnosis, or MAL-ID, to kind of parse these data, build a model, and predict diseases, predict disease and interpret models. So, they were looking squarely at BCRs and TCRs. So thinking about this paper and thinking about their approach, they use blood samples, but I mean, couldn't you use something similar coming into the T1D TCR BCR Air Data Commons repository, and pull out some novel findings using AI and ML at this point?
Yeah, no, absolutely. I mean, I think, I mentioned the number of papers that are citing iReceptor and the Air Data Commons as a source for data for machine learning are, I think, show a definite trend in that direction. I think people are trying to reuse data. There are a lot of challenges in using this type of data, and that's mostly around how the data was sequenced and prepared for sequencing, and the type of sequencing protocols used. Quite
So you
often,
mean like whether or not they used adaptive or 10x or whatever?
Exactly, because what you get is people will show that you tend to get a lot of signal in your AI due to the type of protocols used, essentially. And so that's one challenge. And the other challenge, and this is something that we're actively working on solving. One of the challenges of AirSeq data for machine learning, or one of the gaps, is that most of the sequence data in the Air Data Commons. So there's 5.2 billion sequences annotated with VDJ annotations, CDR3, 1, 2, and 3, CDR1, 2, and 3. So a very, very rich data set. Almost none of that data has any information about which epitopes those receptors are specific to.
Right.
And that is something we're actively working on solving. Some data in the Air Data Commons does have epitope information, epitope reactivity information with it. And I think once we pass that threshold, I think the data will become much more valuable for that, because I think there'll be much more signal in the data when you know that a repertoire of 1 million sequences has this percentage of receptors that are known to have some sort of reactivity to an epitope, and which epitopes and which diseases and which antigens those are specific to. So I think there's, I think the data is valuable as it is, because and I think that's indicated by all the citations. But many of those papers say, well, it would sure be nice if we had epitope specificity information with these receptors.
Yeah, I definitely I've heard there's a buzz on the street that that's where people are trying to go next, for sure.
Yeah, and I believe the Boyd paper, certainly, I think they actually cite iReceptor as a source for data
I think that's right.
in
Yeah.
this context. I know there's a Zaslavsky paper that does that. I don't know if it's this one for sure. A 2025 paper from this group. So this might be the one, I can't remember, but yeah. So they're aware of, and they comment on it. I don't know what they say about it. I can't remember off the top of my head, but
I think it was good.
They also, I mean, I know they mentioned that they stored their data in MyAir compliant format. Then they didn't give a reference, which was kind of odd, but I assume that's like the data, not the metadata, but it's a good step forward, really.
Felix and or Brian, can you guys just comment on the importance of metadata in these kinds of studies, and how sort of the bright spot that Air Data Commons iReceptor is in handling metadata that comes with all of these data?
yeah, I mean, that is the power of the Air Data Commons. So remember, back in 2015, the Air community came together, and the big thing they did at the very, very beginning, the first paper from the Air community, other than the one that says established the Air community, was the MyAir standard, the minimal standard for air studies. And it basically describes on the order of 80 or 90 fields that it recommends researchers consider gathering for their studies. And that data is in a standards compliant data format. So you can basically compare apples to apples and oranges to oranges. So when you are querying a repository for age or species or disease diagnosis, you know that all of the 5.2 billion sequences have metadata that describe them, saying that this sequence, you can identify this sequence comes from a subject with this disease diagnosis. And that's a very robust. And without being able to do that in a robust way, you really can't use the data all that effectively. So that's really, really critical.
And let me just ask you, what other data repositories have that capacity or capability?
So a lot of repositories store that information, but it's not in a way that you can actually compare it to another repository, right? So you might have age, but it might be in a text field, and the number and the unit in 10 and years might be just stored as a text string, and it might be 10Y, it might be 10YRS, it might be 10YR, it might just be 10, right? And so without that, the ability to do that in a computable way, you can't really compute on the data. So most repositories would have metadata of some kind, but it's typically not very useful.
So back in 2022, when my friend pointed me in the direction of the Air Data Commons, saying that that was one of the biggest features, most important features, and that holds true today. So I just wanted to kind of highlight the uniqueness of it. And I guess I would also say lastly that It does have this really, I know you tapped on this, but I'm just going to reiterate the fact that as a researcher, and you, especially maybe you're an early career researcher, or you have this very hot hypothesis, you can actually load your data in to, your own part of the repository. Keep it silent, keep it dark, basically, as you compare it to the data that's already in there. Then when you're ready to publish your paper, or the paper has been accepted, you basically just hit hit send, and your data is then fully released, very easily released into the repository. So I think that that's just a really unique feature that is such a time saver. I mean, most people are, once the paper is published, they're scrambling to get their data all in line and open under their NIH guidelines for open data. But, if you've already been sort of, putting a little away every day or every week, and, at the end, it's just a lot easier just to press that button and release it into the repository. And plus you have the added benefit of earlier access, using your data as a lens to look through what's already been deposited. So it's really valuable.
Yeah, to that point, Monica. So I was a little bit cagey. I don't know if people noticed it, but I said we're approaching 400 million sequences.
Yeah.
One of the reasons for that is, so this study, this is a really, really interesting study. It's from Todd Brusco's lab. It's basically 300 million sequences in itself, from 2,000 subjects.
Yeah.
So this is a massive T1D study. And it's been published as a preprint. The data has been loaded. It's sitting in an Air Data Commons repository, but it's not in production yet because the data is under embargo until the publication happens.
Right.
And so we've done the work to do it. It's available. The researchers can use that repository to research, to explore the data themselves if they want. But it's not available to the public yet. And so I've got this being published sometime in the next couple months, because this is in the future. Not
So
sure people
this is a
noticed that.
little, yeah, so this is
So
a
this
sort
is
of like
this is coming soon.
keep
Yes.
checking in, because this is going to
Yes.
be just a mother load of data once it drops.
Exactly. And Erin Michelle's group has another very, very large study that I think over probably maybe will be available about the same time this one comes out of embargo. And I think that's on the order of a couple 100 million sequences as well. So I would expect type 1 diabetes to pass COVID-19 as the largest disease diagnosis data set in the Air Data Commons, probably in the not too distant future.
Yeah.
So.
And I would also add, the role of the sugar science, as well as sort of initiating this whole project and then facilitating its growth. We kind of went out to the top scientists and really convinced them to drop their data in. So we have scientists in this repository, such as you mentioned, Todd Briscoe, Aaron Michaels, but also Maki Nakayama, Sally Kent, Nina Luning-Prack, you mentioned, Iria Gomez, Eddie James. I mean, these are the top scientists. Iria came out of Mark Peakman's lab, that are working in the TCR field right now. So you've got some very, very detailed and interesting data there. And I would encourage those who have interest in these types of data to really either contact Brian, or, just log on and really sort of, tool around in the sandbox there, because there's so much to offer. They've built, Brian really, and his colleagues have built a beautiful repository. Still there, Brian?
Yep.
So,
Yep.
yeah,
Good.
I guess I'd add too that these disease-specific repositories are really important to kind of start focusing and getting people excited that, like Brian put up there, the network effect, but in the whole world, maybe not, but if we could get, certain diseases, no one is going to really publish in type 1 diabetes AIRR without getting towards, getting into our repository and stuff like that. It's a way to make some real progress.
Yeah, for sure it is. So, well, thank you both for spearheading this enterprise and, actually technically putting it all together, and for Felix for being the brainchild of the entire AIR Data Commons. So, thank you both, and we encourage everyone, come one, come all, and have a look at what this really great tool has to offer. All right,
Well,
with that,
thank you, Monica, for bringing us together.
yeah,
Thanks a
for
lot.
sure. All right, have a great rest of your day, everyone. Bye-bye.
Okay, bye.
Thank you. See ya.